Good afternoon, everybody, in the lecture room and also online. Thanks for coming. It's my great pleasure to welcome Vladimír Křen to give us a presentation of the recent paper about nitrogen-containing flavonoids and bacterial resistance as the first presentation of the INTERMICRO seminar series. Which I hope will be a successful one, and helping us to also learn something from the others, so that we can utilize the project in its best. Thank you very much, Vladimír, for taking this challenge, and the floor is yours.
So thank you very much, Jan, for a nice introduction, and it's a big pleasure, and I'm really happy to open this series, which definitely will be successful and very stimulating. So today I'm going to talk about part of our research area, which is performed by our so-called yellow team, which deals with natural products and mostly flavonoids, which are by nature yellow. So that's why it is yellow team. I must say that most of the work is the work of Martina Hurtová, who is now expecting a baby, but who did most of the chemistry, and most of the microbiology has been done by Jitka Viktorová from the University of Chemical Technology, if I'm right. So I'm here like just a presenter and coordinator of all these joint efforts. Anyway, this is the paper we are going to talk. The good thing about this publication is that we have tokens for open access at the American Chemical Society, and this is quite a decent paper from, I think this is quartile 1. So it's not as bad, although the chemistry paper have the generally lower impact factor, but this is a general feature of the chemistry journals. Anyway, let's go to the microbial resistance. This is a plague of contemporary medicine, and everybody knows that this is a big problem and also major challenge for this century. Not only in the antibiotics therapy and treatment, but also in the cancer chemotherapy. And it is very well known that most of the pharma companies are not happy and not much keen to develop a new antibiotics because it is not much profitable. So we have to deal or handle all the infection with older antibiotics, and the multi-drug resistance is developing. I think you all know the basic mechanisms of this phenomenon, which is partly mediated by transmembrane transporters, but also modifiers, both on the side of antibiotics and target proteins. As I said in the intro, we are dealing with flavonoids, and there's a lot of flavonoids that has been already identified, like quite efficient multi-drug resistance inhibitors. Here there are some examples of existing basic compounds which are occurring also in the natural products in the resources. For instance, epigallocatechin gallate, bicalain, naringenin, and taflavin in tea. Okay, but the activity of this compound is not much great. So we have started to perform modification of these compounds since maybe 10 years ago, and that's the results I'm going to present to you now. Flavonoids is a group of a great number of compounds of various structures. There's over, I would say, 3,000 compounds maybe, and here there are some basic structures, so I'm not going to go in much details in the chemistry, but there's much more than what is here shown, and these are just the basic skeletons of flavonoids. This research actually started with pure serendipity. last week there was a very nice lecture of Morten Meldal. That's the Nobelist from, I think, 2022 in the Institute of Organic Chemistry. And he sent very clear message. 95% of all Nobel Prizes are based on the serendipity. Never plan your Nobel Prize. And that was his message. And this is exactly what happened here. This is actually a new reaction described by Martina Hurtová, who was purely serendipitous, because my original idea was something like synthesis of this compound by conjugation of dibromosynamic acid with some other compounds, but this reaction didn't work. What worked, however, was another reaction, and it was halogenation, or in this case, bromination of the target compounds. So this structure, or this dibromohydrosuccinic acid, proved to be excellent brominating agent, so far undescribed. And that led us actually to the series of very nice reactions compared to another halogenation reactions like bromine. It's no way, because it is oxidizing and making very complex mixture. This brominating agents normally used in organic chemistry gives a mixture of products. Another brominating agents, again, mixture of products, and even after longer reaction time, over-bromination. So this really was a great reaction. We call it Hurta reaction, according Martina Hurtová. And this actually led us to synthesis of series of halogenated flavonoids. We have prepared a series of not only brominated, but also iodinated and chlorinated flavonoids. So that was the first start of the whole story. We synthesized something like 50 new derivatives, and we decided that we will continue in testing them in biological experiments. Halogen derivatives are very useful synthons for organic chemistry. Here you see a survey of various reactions where you can start from halogenated aromatic compound. And the first reaction we have tested was Suzuki cross-coupling with boronate acid. I'm not going in details in this chemistry, but this is really nice chemistry. So it allows you to modify flavonoid skeleton with another aromatics bearing even fluorine, which is always a very much desired derivative in as a pharmacophore. And also this CB bonds, boronated structures, which are another synthons for further reactions. So here you see the Suzuki cross-coupling reaction using these boronates with this fancy catalyst, giving you this allyl and other derivatives, and also cyclic stuff. And actually, we found that some of these cyclic flavonoids are found also in the nature, although they're very rare. So the synthetic procedure leading to them is very helpful. So, okay, this is all the derivatii, and this is the first series of derivatives which were prepared in our group in 2022. And you see a lot of new derivatives on flavonoids based on mostly quercetin. So far, so good. But then we wanted also to prepare nitrogen-containing flavonoids, because nitrogen is very potent pharmacophore in also occurring in the multidrug resistance inhibitors. So here we needed to introduce the CN bond in aromatic compounds or aromatic amines using this reaction. And for the optimization, we needed to optimize temperature catalyst and also ligands. So here there are some examples of known flavonoids which are containing nitrogen, which are partly found in nature, partly as a semi-synthetic compounds. And these compounds were kind of inspiration for us for synthesizing of new derivatives. Here, we decided to go for CNC aryl alkyl aminoflavonoids, this basic structure, and again the same scheme, but here we didn't use, as previously, Suzuki cross-coupling, but Buchwald-Hartwig reaction, which actually is leading to the nitrogenous derivatives here. And here's the chemistry. It is using palladium as a catalyst, metal palladium, and starting again from the halogenated substrates from the previous part of our work. And here there is a whole series of optimization and the whole play. It is not quite trivial, but eventually we managed to prepare again the whole series of nitrogen-containing flavonoids based on flavon, on quercetin, and on luteolin. And then we went to biological testing. First, when you test any new compound, you have to test the cytotoxicity, because what is cytotoxic? There is no way to continue further with any further tests. And we found that fortunately the cytotoxicity is in a good level. So in all cases, in all compounds, it is over 25 micromolar, which is very well over the benchmark doxorubicin. Antioxidant activity, that was quite interesting, because flavonoids are quite well known for antioxidant activity, although here the antioxidant activity was somehow decreased, which is not the issue, because actually we are not seeking new antioxidants. And the same was for anti-inflammatory activity. Some compounds, some parental compounds possess anti-inflammatory activity, but here in our case, most of the amino acids decrease anti-inflammatory activity. But generally, all these results open the way for further testing, because we are interested in the multi-drug resistance. So the main thing is the low cytotoxicity. Good. And here are some results already for the multi-drug resistance inhibition. We used first colistin-resistant Pseudomonas aeruginosa, and we tested the minimum inhibitory concentration. Actually, what is basic kind of request for any inhibitor of multi-drug resistance is that this compound should not have by itself antibiotic activity. Because when you have some compound with antibiotic activity, then it could develop again resistance. So the compound should not have by itself antibiotic activity, which was the case. So that was okay. So by addition of these compounds in various concentration, we managed to restore or reverse resistant phenotype in colistin resistant Pseudomonas aeruginosa. Here you see two most efficient compounds. Please notice that there is always fluorine in the compounds here, trifluoromethyl, and here as well. And then we tested also gentamicin resistant Staphylococcus aureus. I'm going to go in details later on. And also in this case, we identified three compounds, definitely better than the parental compounds. You see always the derivatives are better than the parental compounds, which are marked here by black lines. And the concentration of these compounds restoring or reversing resistant phenotype is well under 10 micromolar. So far, so good against fluorinated compounds. So the fluorine is obviously important as a pharmacophore. Then we tested also more strains. These are actually the clinical isolates. Clinical isolates, real pathogens which have different resistant genes here. the ERMA and C genes, which are encoding for ribosomal methyltransferase, so modifying ribosome as a mechanism of the inhibition. And you see that with this compound, with this trifluoroaniline quercetin, we obtained nearly 5 times better results than the parental compounds and synergical effect with the antibiotics. So I would say most of the new derivatives were more potent than the parent compounds, and the therapeutic indices have increased significantly when considering the toxicity, the low toxicity. This is just the mechanism how it happens. The methyltransferase is simply methylating adenine or adenosine using some and modifying thus the I think 30S subunit. So this actually confers the antibiotic resistance. So molecule of erythromycin is here. So we were able also to kind of analyze and determine the mechanism of this inhibition or the elimination of resistance. Here there is some molecular modeling and docking of the compounds into active site of the methyltransferase, and it's quite puzzling pictures. You don't have to go in details, but what is most important that the docking results show that the most efficient compound is docked very well in the active site, kind of replacing some cysteine in the pocket and forming very dense network of hydrogen bonds. And this is most plausible mechanism of the inhibition of the adenine methylation activity. So we were not able to we were able to find the effective compound, but also to determine the mechanism of its activity. Then there was another series of experiments targeted to the gentamicin resistant Streptococcus aureus, and here are just the targets on the molecule of kanamycin, the modification of the antibiotics. So this is different way of resistance, antibiotic modification. So we had two resistant strains which were positive for the modifiers or this is actually phosphotransferases modifying the molecules of antibiotics. And here are some results. So again, trifluoromethyl derivatives was the best, showing synergic effect with gentamicin. And surprisingly, we actually found that these strains or the gentamicin is not the target for this phosphotransferase. So we thought that the mechanism might be possibly modification of the gentamicin, but anyway, this is probably not quite clear and not definite mechanism, because we have also tested or incubated gentamicin with this phosphotransferase, and we didn't find any reaction product on that. So we don't know yet what is exact mechanism in this case, but the point is that the results, the antibiotics inverting activity is relatively high. So these compounds could be kind of or they have a good potential to be used as an adjunct therapy with the antibiotics, also thanks to their very low toxicity. So going to conclusions already, the introduction of aniline groups at the C8 of quercetin eliminated the anti-inflammatory activity, but maintained low cytotoxicity, which is important. Some derivatives of the quercetin and also luteolin modulated erythromycin resistance in Staphylococcus aureus by inhibition of ribosomal methyltransferase. And they also, I didn't mention that, but they also acted as a transmembrane efflux pump inhibitors at lower concentration. And we had also effective modulation of gentamicin resistance in Staphylococcus aureus, although the exact mechanism remains unknown. And this quite nicely shows the potential of this anilino-substituted flavonoids in combating multi-drug resistance. And our next target or next kind of mission is to prepare or develop a fluorination of the flavonoids, because nobody so far managed to perform or to accomplish direct fluorination of the flavonoid skeleton. This is really a challenge. We have some ideas how to do that. A lot of reaction proved to be unsuccessful, but I hope in 1 or 2 years, thanks to our excellent chemist we have in our group, there will be some nice paper presented again in this project. And with that, I would like to thanks to my co-workers, mostly Martina Hurtová, who did most of the chemistry. Then the people from Institute of Organic Chemistry for structure identification, and Jitka Viktorová and her team who worked on the antibiotic resistance analysis. And to you, I thank you for kind attendance.
Thank you very much, Vladimír.